Hi, thanks for tuning in. This video contains a segment from the NNLM Training Office August 2017 Teaching Topics session, where we had 7 fantastic medical librarians share 5 of their successes, flops, and far-out ideas. Check the description below for links to the other videos from this session. In this video, we will be hearing from Michelle Price, who is a librarian at St. John Fisher College. She's going to talk about using a flipped class model and employing group work when teaching PubMed and Gene to biology students.
Hi, welcome. My name is Michelle Price, and I'm a librarian here at St. John Fisher College. We are a doctorate granting institution, but we have a large undergraduate base. There are 9 professional librarians that work at Library, and in my role, I'm the liaison to the Wegman School of Nursing, the Wegman School of Pharmacy, and for those people who like to grocery shop, yes, it is that Wegmans. And the departments of biology, chemistry, and physics. Today I will be discussing the first part of a 2-part lesson for the cell biology lab. For more details on this lesson, I've included the link to my e-portfolio on the slide, and it's also coming in your chat box. It includes the full lesson plans, worksheets, and assessment materials. On the left of the screen is a curriculum map of library instruction for our biology majors. In addition to a strong first-year program represented in green, library instruction is heavily mapped into the biology program. This also includes organic chemistry, here in orange, that all biology majors must take. A steady stream of skills is introduced, reinforced, and emphasized almost each semester. The biology majors are very used to seeing a librarian, and in particular, me in their class. This exposure prepares them for the learning objectives for cell biology that are listed to the right. You can see here that students will describe NCBI and its family of databases, and specifically in this class, how they relate to Wormbase, a model organism database outside of NCBI. Then, the second objective, we go to work searching in and across databases. I wanted to address a major part in constructing and delivering this lesson. When the cell biology lab coordinator first approached me about working together, we weren't quite sure where we were headed. My first attempt felt awesome, complete, exposing students to NCBI and the story of data, and it was a flop. You can see here on the left-hand side a slide from that presentation. It was too broad, and although accurate, it didn't connect deeply enough to the lab, and it didn't connect to the final paper and presentation that students had to prepare on a gene. So how did I fix it? I must also add at this point too that my undergrad is in Spanish. My master's degrees are in library science and education, so I did not have the content knowledge to address the science topic. So how did I fix it? I spent a summer in the lab doing pieces of lab activities and working with other students conducting research in the lab. This gave me a better understanding of the world of cell and how it feels to work in a lab setting and search for information in that context. In the fall of 2016, I took a 6th contact hour course from NNLM called Chemicals, Drugs, and Genetics, Oh My! Searching PubMed and Beyond, which covered many things but brought me deeper into the gene database. Finally, I attended several sessions of the students' final presentations. From there, I was able to revise the lesson, as you'll see on the next screen. Each section of the lab has to complete pre-lab work with the quiz before I meet with them. I'm added as an instructor to each Blackboard section of the lab, and I can add materials and view quiz results. I rely heavily on the expectation that students will complete their pre-work lab before every quiz, including the one that I run. And so far, only a few students have come unprepared for class. Their pre-class quiz grade does feed into the grade center, and it does count towards their final grade. Flipping the class in this manner does mean that I must be willing to adjust my teaching on the fly. For example, if Lab A is having trouble with something in PubMed, I need to address it before their group activities. With the mechanics covered in pre-lab work, class time is spent on application, which is exciting, but it requires different teaching techniques and a different mindset. There is more variability, and the class activities have to be designed to elicit higher order thinking. So in the spring of 2017, after my course, I added Gene to the instruction. This created the opportunity for students to spend more time on the finer points of PubMed and Gene and understand the relationship between the two databases. As you can see on the slide, on the left is a screenshot of a search for PSIN2 in PubMed. It translates to an all-field search, and a box appears at the top of the results with the suggestions for Gene. The same search on the right in Gene will limit to species by the nature of the record, as you see under the description. It gathers all the aliases in the search, and then the bibliography then leads you into PubMed. There is a lot going on in just these few screenshots. How this all comes alive for students is in the group activities. The students are already in lab groups, so I asked them to stay with their lab group and give it a name. On this last slide, you will see 2 group activity questions and the student answers up on the board. All group members must agree on the strategy to put up on the board. Then as a class, we compare strategies and techniques. Students are often amazed that their search for something can lead to such a radically different set and number of results compared to their peers. For example, following the blue arrows on the bottom of the screen, the results differed from 4 to 117 results. Now this lab section had 3 groups, and from their posted answers, we were able to discuss many different topics, but they all arise from their answers. They are not something I prepared and handed to them. For instance, given the examples on the board, we talked about when to use subject headings and limiters versus free text or keywords, words that have synonyms and related terms like men and male, when to use a date limiter, gene names and the use of the word gene, the effects of quotations on a search, what the full text limiter in PubMed does, gene names and aliases, and the human limiter versus the bibliography link on the Homo sapiens record in Gene. So ending on that last slide and moving to questions. If there's any questions, I'll look in the chat box.
Here we go. We have one from Kate. Did you create your own videos or did you reuse those from NLM?
So the 5 videos in the PubMed PreLab Work, only one of those did I create on my own. The rest were from NLM, and then the Gene video was from NLM as well.
Great, thank you. I would like to just ask, what's one piece of advice you'd give someone watching today about successes, flops, and far-out ideas for teaching PubMed?
Be willing to revise your session, including spending a little time understanding the content a little better, the needs of your students, and then be willing to apply different pedagogical techniques in your revision.
Awesome. Thank you.
Thanks for watching. This video was produced by the National Network of Libraries of Medicine. Select the circular channel icon to subscribe to our channel. Select a video thumbnail to watch another video from the channel.